Okay, Assalamualaikum warahmatullahi wabarakatuh. Finally, we are here at the topic of translation, the final subtopic for the chapter 4. My name is FATHIAH ABDULLAH and you can contact me through my email fathiah@uitm.edu.my. So before we start, let me first explain about the picture that I've put here. If you can see, there's a thread there showing that is about elongation of a stitch. Okay, so this also represent the translation process. We have on the slide there the ribosome, the mRNA strand, the tRNA, and the newly formed growing protein. Genetic code will be later translated into a polypeptide. Okay, so what is genetic code? Genetic code is almost universal in all organisms that we have on Earth, and the code is non-overlapping groups of 3 mRNA nucleotide, and this non-overlapping group of 3 mRNA nucleotide is known as codon. And there will be no space separating the neighboring codon, and the codon is a triplet basis on the mRNA strand. Okay, again a triplet basis on the mRNA strand. So we have about 64 possible codon with only 20 amino acid coming from only 4 base, which is U, C, A and G. And not all amino acids have equal number of codon coding for it. For example, let's have a look at the table at your right side there. For the tryptophan, or simply said as TRP, they just have one codon, which is the UGG. And for the leucine, they have 6 codons that determined by the leucine, which is UUA, UUG, CUU, CUC, CUA, and CUG. So that's how we read the table. We start with the left one, the first mRNA base. Normally, it will located at the 5' end of the codon. And the second mRNA base, which is in the middle one. And the last one is the third mRNA base at the 3' end. So let's say if I ask you what is the methionine, alright, the green box there, which is their coding for AUG. So meth is coming from the AUG, or the codon, the specific codon for methionine is AUG only. There are 2 groups of codon that you are compulsory to remember, which is start codon and stop codon. So, start codon is represented by the AUG that corresponding to amino acid methionine. And the stop codon, you have 3 codon for this stop, which is UAG, UAA, and UGA. Later on, this start codon and this stop codon will be very, very important in the translation process. Okay, so in the tables there, we have the 20 amino acid. Okay, let's say its abbreviation is ALA or ALA. The full name for that group is alanine. ARG for arginine, and so on until you have the VAL, which is for valine. And what is anticodon? Anticodon is a sequence of triplet based on tRNA that is complementary to and combined with codon of mRNA. So these are all the codons on the mRNA, but for anticodon, it's a sequence of triplet also, but it is situated at the tRNA. Later, I will explain to you. So the translation process is a process of translated the mRNA into a protein. From genetic code will be translated into a protein code. So we started everything with the DNA strand, then they've been transcribed into RNA, and then when you do have the mRNA, it will be translated later to form the polypeptide. That polypeptide is basically coming from the arrangement of lots of amino acids. Okay, Francis Crick, okay, the scientist name that we have discovered during our DNA replication, has proposed a theorem we call as a central dogma. Okay, central dogma is basically how you have the DNA and then later on producing RNA and later on will producing protein. And the association between DNA, RNA and protein at the molecular level can be shown as example here, where you have the DNA template starting with the ACC, AAA, CCG and AGT. Later on, you will produce mRNA with UGG, which is notice that U is transcribed from A, G transcribed from C, G transcribed from C, and same things happen. Okay, U transcribed from all A up there, GGC transcribed from CCG, and UCA transcribed from AGT. So this mRNA here is a complementary to DNA template. Alright, so here you can see, also look at the end of each templates, end of each DNA and mRNA. DNA they started with the 3', mRNA with the 5', knowing that the new nucleotide that form in the transcription process will be added at the 3'. So the starting point for the mRNA will be at the 5'. Okay, later on, after you do the translation process, you will have a tryptophan, which is a amino acid from the UGG, okay, phenylalanine, which is the amino acid for the codon UUU, glycine is the amino acid for the codon GGC, serine is the amino acid for the codon UCA, and etc, etc, etc. So this protein will be go on and on and on until they found the stop codon. So the string of amino acids has a direct relationship to nucleotide bases in RNA and DNA. So any mistake that happen in DNA will create some error in the mRNA and also will create another error in the protein. So translation is the production of protein from a mRNA template. This process will involve tRNA, codon, anticodon, and ribosomes. And ribosomes make protein by pairing the tRNA and mRNA, catalyze a condensation reaction between the ends of the amino acid to make the peptide bond, using the peptidyl transferase activity. Okay, this peptidyl transferase activity is carried out by rRNA, and the protein is released either to the cytoplasm or to the endoplasmic reticulum in eukaryote. This is a unit structure of tRNA. So the function of tRNA is to transfer amino acids from the cytoplasmic pool of amino acid to a ribosome. So how do they work? Okay, first of all, let's have a look at the right side of our tRNA there. We have one is the one with the red box is the tRNA that carrying serine, and with the green box is the tRNA that carrying tyrosine. Notice that at the serine, they attach that particular tRNA are attaching at the codon code AGU. And what is the anticodon? Anticodon is the codon that been carried by the tRNA. So basically, if you have a single mRNA wandering around in the cytoplasm, and then you have the AGU codon, the types of tRNA that will come and bind to the AGU are the one that are carry the amino acid serine. Okay, so meaning that the one or the codon that are attached to that can attach to that kind of AGU codon is the anticodon UCA. Again, okay, for the tyrosine, let's say, if you have a single mRNA wandering around with the UAC code, which is that UAC, if you look back at the codon table, UAC is the codon for the tyrosine. So any tRNA that carrying tyrosine will come and attach at the UAC at the mRNA. Okay, so let's have the left tRNA that are more colorful there. We have a structure of tRNA there. You have two types of RNA. One is mRNA and the other one is tRNA. So mRNA code, again, let us read it from the 5' to 3'. It has a codon GCC. So the anticodon that will come here is the one who bring the amino acid of GCC. If you refer to the codon table, this GCC is actually an amino acid alanine. So the tRNA that are carrying the alanine will come and bind to the codon GCC. And the code they are complementing with the GCC codon is the anticodon CGG, meaning that here you will read mRNA codon from 5' to 3', but the anticodon will be read from 3' to 5', which is here is the CGG. Okay, since day one, we know that ribosome is something that produces protein. Okay, so the ribosome will add each amino acid that been brought to them by tRNA to the growing end of a polypeptide chain. And this mRNA will attach at the small subunit of ribosome. So ribosome is basically comprised of a large subunit and a small subunit. So when they combine together, they will form a ribosome. So at the large subunit of ribosome, we have three sites: the E site or the exit site, the P site or the peptidyl tRNA binding site, and the A site or the amino acyl tRNA binding site. And the lower part at the small subunit, we have the mRNA binding site. Okay, similar as transcription, translation also can be divided into three main stages, which is initiation, elongation, and termination. For the first step, initiation, we can see that these are all the components to start the translation, where the mRNA will bind to small ribosomal subunit first. You can see that you have a single strand of mRNA and you have a small ribosomal subunit down there. Okay, and initiated tRNA with complementary anticodon to start codon AUG binds to mRNA. Okay, you see the blue colored one, initiated tRNA. That initiated tRNA are carrying the amino acid methionine, and they complement to the start codon AUG. So the initiated tRNA will come first, bind to the start codon. So this tRNA amino acid carries the amino acid methionine. Okay, have a look at the complementation or complementary of the codon and anticodon there. You have the codon 5' AUG and the anticodon is the 3' UAC. Okay, so when you have the methionine, then comes the large ribosomal subunit to complete the initiation complex. This large ribosomal subunit will carry the P site and the A site. Okay, the initiated tRNA is in the P site, which is the middle. The A site is available to the tRNA bearing the next amino acid. In this process here, one phosphate will be used, and this phosphate is coming from the guanosine triphosphate and will form guanosine diphosphate. For the elongation part, we can divide this stage into codon recognition, peptide bond formation, and translocation. Okay, the elongation cycle of translation will be continue after you have started or after you have initiated the translation. So here, let's have a look at the ribosome subunit at the beginning of the slide or at the upper side of the slide. We have a smaller subunit, we have a larger subunit, we have the EPA site. Notice that at the P site, there's already a tRNA containing 4 amino acid. Okay, now here comes the new tRNA with his own amino acid. They'll come and bind or fill up the A site. So this incoming amino acid tRNA will fill up the A site. After that, the ribosome will catalyze the formation of peptide bond between the new amino acid and the carboxyl end of the growing polypeptide. It's kind of all the amino acid or the amino acid group that has been formed at the P site will kind of jump into the amino acid that newly come, which is at the A site. Okay, so the amino acid from P site will transfer onto amino acid tRNA in the A site. Okay, then at this stage the ribosomes move one codon down mRNA. So the one who move is the ribosomes. So the tRNA with growing polypeptide chain in the A site is again translocated to the P site. Meanwhile, tRNA in the P site move to the E site and is released from the ribosomes. So that's why they are named as the E site or exit site. So here, the empty tRNA at the E site will go out from the ribosome and will find another amino acid. Okay, and the cycle will keep on continue to prolong the polypeptide. Okay, for the termination process, when a ribosomal reaches a stop codon on mRNA, the A site of the ribosome accepts the release factor. So here, the stop codon will be detected at the mRNA strand. So when there comes the release factor, this release factor will hydrolyze the bond between the tRNA in the P site and the last amino acid of the polypeptide chain. So the two ribosomal subunits and the other components which are polypeptide chain, mRNA and tRNA of the assembly will dissociate. We know that for the endoplasmic reticulum, we have two types of endoplasmic reticulum, which is the smooth ER and also the rough ER. So here is a little bit about how you form the rough ER. Okay, these are on how the polypeptide from ribosome bring about to the ER to make it rough. Okay, so you already have the translation process whereby you are using mRNA strand to produce or to translate the mRNA information into a polypeptide. So you have the signal peptide at the ribosome, and here comes the signal recognition particle, binds to the signal peptide, bringing the ribosome to the surface of the ER, to the ER membrane, and this SRP here will open up, will attach to the SRP receptor protein and will open up the channel, and then the polypeptide will be sending out of the ER, going to the outside where the polypeptide will be released at the ER lumen. Polysome is a condition when the cell rapidly make many protein. So here many ribosome will read the same message structure known as polysome. So you can see that with a similar mRNA, different or a lots of ribosome actually producing the similar protein using the single strand of mRNA. So finally, we are done with the transcription and translation. Bear in mind, both process do involve the three substages, which is initiation, elongation, and termination. Bear in mind also, transcription has to happen in the nucleus because they are using DNA as a template. You know that DNA can only be found in the nucleolus of the nucleus. Okay, and then here, after you get the mRNA, that mRNA have to go out to find the ribosome because you need ribosome, the EPA site in the ribosome. So mRNA will come out or will go out from the nucleus, and finally you are using the same mRNA to translate the information that you have in the mRNA to form the polypeptide. And also this polypeptide is coming from lots of amino acid. Okay, still remember the 20 groups of amino acid that coming from the only 4 base, which is A, C, U and G. Okay, so to answer our learning outcome for this protein synthesis, can you explain the translation in protein synthesis? I hope you can. Bye.